We'll finish up Module 2 with a discussion of how diffusion occurs through a cell membrane. Okay, basically you have to understand that a cell membrane is a semi-permeable membrane. Some things can pass through, some things cannot. Sometimes it depends on the condition of the cell whether things can pass through or not. But what I want to divide these substances into is these four groups. First of all, lipids. Lipids can move freely through the lipid bilayer. Because lipids, oil mixes with oil very nicely, and so the lipids can move directly through the phospholipid bilayer. In fact, many anesthetics are lipids because they can move into cells quickly and alter the cell sensitivity. Secondly, the gases, specifically oxygen and carbon dioxide, which have to move through the cell membrane of all living things, these also can move freely through the lipid bilayer. Now we'll move on to water and small ions. These move through channel proteins, and we'll discuss those a little bit later, but those are one of the many types of proteins that are embedded in that phospholipid bilayer. Water, in fact, goes through very specialized protein channels called water pores. Specifically, they're called aquaporins. Your book doesn't mention these, but I'll tell you a little bit more about those in a little while. Large molecules, even things that we don't think of as that large, like glucose, cannot pass through the cell membrane except where there are very specialized proteins called carrier proteins. And again, in the following pages, we'll go over those as well. This diagram is from a different textbook. It's a little more specific than yours, so I wanted to go ahead and show this one to you. It's a Benjamin Cummings diagram that I have in your note set, and this refers to the previous page. Number 1, lipid-soluble molecules can go right through, okay? The gases were not included, so I added those, oxygen and carbon dioxide right here. These can move directly through the phospholipid bilayer. Number 3, we see an ion here. We don't know what it is, but I think those others are representing water. And this shows them going through channel proteins. Again, it's a specialized embedded protein molecule in that phospholipid bilayer. In this case, it's a channel, not gated, but sometimes they can actually have gates and open and close. Then down here, number 4, large molecules, and I'm assuming that this may be some sort of a protein here. This may be a glucose molecule. These cannot go through unless, again, there is some sort of a specialized protein molecule, such as a carrier protein. And we'll get through those in a few minutes. This is the picture in your textbook. It's showing here that gases, such as oxygen and carbon dioxide, small nonpolar ions, such as lipids, and this is where the confusion is. It also says water can cross the bilayer freely. Most textbooks say, and the consensus is, that there are specialized protein molecules, the aquaporins, that allow water to go through a cell membrane. What this really means is, I'm not going to ask it on a test. If there's controversy, let's just not even talk about it. But I think, excuse me, the consensus is that there is a small specialized protein molecule through which the water travels. Other solutes, as it says, cannot cross on their own. We have the X here, and the next page shows the protein molecules that do allow these substances to go through. Again, in your textbook, it's showing here some things can go directly through. Again, when we're talking about gases and lipids and things like this. We also have 2 different types of mechanisms, one called passive transport, one called active transport. Passive transport has some specific criteria. It's generally a channel protein, which may or may not be gated, but in this case, it allows for substances to go through with the concentration gradient. So again, whatever these little green balls are supposed to represent, there are more of them out here than there are inside, and so it moves with the concentration gradient. No energy is required. Over here, in this situation, we have a little bit more complex protein molecule here, and this is called a carrier protein. And in this case, there are a couple of different things going on. First of all, whatever's in this little blue vesicle, it is being pumped through this ion, whatever it may be, against the concentration gradient. Okay? The other thing, because it is going against the concentration gradient, ATP is required, energy in the form of ATP. So that's why this one is called active transport. Energy is required. Okay, so once again, section 5.7 in your textbook, some molecules, particularly the larger ones, have to have these transport proteins. Passive transport is with the concentration gradient, no energy required, and it involves some sort of a channel protein, may or may not be gated, which means it can open or close. And some examples of those might be glucose or an amino acid, something like this. The active transport, remember, active means energy is required. So this involves a carrier protein, a little bit different than our channel protein. This one, I equate it to a revolving door, kind of a silly analogy, but I'll show you why in just a few minutes. The big deal here is it's against the concentration gradient and energy is required. We're going to look specifically in a few minutes at a type of active transport system called the sodium-potassium pump, which is common in many, many cells of many different living things. Okay, passive transport, also called facilitated diffusion because it is with the concentration gradient, only it's being facilitated or helped by that protein molecule. In this example, we have what appears to be sugars coming in and attaching, and then this happens to be a gated one. Then it opens up and the glucose goes on into the other side. Again, with the concentration gradient, no energy required. This is the same deal from your textbook. We have glucose working its way through this. It's changing shape slightly as the glucose comes in and then is spit out on the other side. Once again, no energy required, and it is with the concentration gradient. So this is passive transport with a channel protein. Active transport, I'm going to show you several different images of this, but this is a really important one, and so it's a little more complex to get a hold of, but I know you can do it. I know you can do it. Okay, so again, here's our phospholipid bilayer, and this right here, this blue thing, is our protein. That's our active transport, our carrier protein. And what this is telling you is that energy is going to be involved. It's active transport. Energy is involved, and the movement is against the concentration gradient. Now, what occurs in many living things is that there is a higher concentration of sodium, represented right here by the purple dots. Sodium is abbreviated Na. The symbol is Na for natrium, which is the Latin word for sodium. And it generally is at a higher concentration outside the cell. Inside the cell, we have these orange dots right here, and those are representing potassium. Potassium, I don't know what it stands for, to tell you the truth, but the symbol is a K, okay? Whereas here, the symbol for sodium is Na. And there's a higher concentration of potassium inside the cell, and that is the way we want it, okay? But by diffusion, these sodiums every now and then do slip in, and every now and then these potassiums do slip out. Okay? And so constantly what our cells are doing, they employ this sodium-potassium pump that is constantly working to get the sodiums back where they belong, and on the outside, and the potassiums back in where they belong. Now, this is a crazy analogy, but a student told me this years ago that she had learned this from her psychology instructor, because this sodium-potassium pump is very important in neural transmission, in muscle contraction, in all kinds of things. So it's a fairly standard example of active transport. So anyway, the way she said it, and I'll just throw it out there. Your cells are like, each cell is like your house. Let's put it that way. Each cell is like your house. And inside your house, you have lots of pots and pans. Okay? So that's these orange potassium for pots. Okay? Outside in your front yard, you have lots of sod for sodium. And that's where you want it. But life goes on, and every now and then, especially if you have kids, you might find your pots and pans out in the front yard. And as people go in and out of the house, you might find some sod in your living room inside your house. You don't want it there. You don't want it there. So what happens is it requires some energy. Someone has to go out there, pick up the pots and pans out of the front yard, and bring them back in right here. Okay. Someone also has to sweep up the floor, get that sod that has snuck in, and take it back outside. Okay. And that individual that does that is the sodium-potassium exchange pump. It requires energy. It is going against the concentration gradient. So that might help you remember that. Sometimes in this case, this is also called co-transport, because two different ions are being moved pretty much at the same time with the same pump. Where they belong. Whoa, what happened there? It has snuck in. Whoops. I have all kinds of pictures that I found of this. Of course, this one upside down from the previous one. So in this case, this is inside the cell. Out here is going to be outside the cell. You still have represented the phospholipid bilayer. This right here is our sodium-potassium pump. And as you can see, in this case, we have the potassiums over here in green, okay? And those potassiums, just like your pots inside your house, every now and then they do sneak out right here, okay? Then you have your sodium, your sod, out in the front yard, but every now and then it sneaks in, okay? So what occurs is the sodium-potassium pump, here's where you can see the resemblance to a revolving door. The sodium will attach to it, the potassium will attach to it on this side. As it changes shape and required by energy, you have the pump moving these items back to where they belong. Okay, so again, this is something else. I'll try to find a video and add this as well to your the page for the second module. So if you want to look at it, also remember you can always Google some of these things. Lots of different views on YouTube of these same sort of lessons. None of this is unique to my class. This is very general information. It's all out there, and there's a lot to be found. So again, if you find some of this stuff hard to grasp, take some time and look at some different YouTubes, and it might really help. Okay, our last section here is on bulk movement in and out of cells. We've been talking really about ions, fairly small molecules that are moving in and out of a cell. But what about bulk movement? This is termed exocytosis, and endocytosis. Exo is like exit, so this is movement out of a cell. Endocytosis means coming in, so this is moving into a cell. Now, phagocytosis is a specific type of endocytosis, and it is sort of important because there are a lot of different cells that do this. Phago means food or solid, and so some cells that do this in humans or in other animals as well, white blood cells undergo phagocytosis. Amoeba undergo phagocytosis by engulfing various pathogens, various food organisms and things like this. So again, these phagocytosis generally involves pseudopods, which we've discussed a little bit earlier. So here's a picture of from your book that's kind of going through endocytosis and exocytosis. Endocytosis, these little red dots, whatever they may be trying to represent, are coming in, okay? Some sort of vesicle is being formed here, and then there are a couple of different pathways here that they're showing. This one as it goes out, and that is being moved out of the cell right here. That is going to be exocytosis. So endocytosis moves substances in, exocytosis moves substances out. The other thing they just added in here is that perhaps some of these vesicles, some of the, as it says, the vesicle contents may be sorted. Some of them here may be waste products, and so they would merge with a lysosome, which has enzymes to help destroy those or recycle those. Others may go into a Golgi body, repackaged, restructured, and then at that point go outside the cell through exocytosis. This is a scanning electron micrograph, very high magnification, of a plasma membrane. These substances have receptors, go to receptor sites, and then a vesicle is formed. And so again, it's going in, so it would be called endocytosis. This picture from another textbook, and I think we've already gone over this, talking about the endomembrane system. But here we have endoplasmic reticulum on the left. This is rough. You see the ribosomes on there, and so that would be producing more likely proteins. Over here we have smooth, so it would be more likely be producing carbs or lipids. Then various transport vesicles are going to bring those to the Golgi body, also called Golgi apparatus, for the final packaging. Then this is showing vesicles doing, depending on what they have in them, some of them may be lysosomes. If they're going to be excreted, they would be secretory vesicles, or they could be membrane renewal vesicles if they merge with and are incorporated into the cell membrane. But again, this is showing exocytosis by means of the secretory vesicles, and this is an actual transmission electron micrograph of that releasing the substance outside the cell. These show some images of phagocytosis. Again, different, in this case we have endocytosis as it goes in, but because it is solid, it could be considered phagocytosis. In this case, we have it merging with a lysosome, which will digest it, and then the waste products would be expelled by exocytosis. And again, this is a scanning electron micrograph of a white blood cell surrounding some sort of pathogen, phagocytosis. And to see this one more time, again, we have the movement here. This vesicles are produced in this case. It looks like these vesicles are coming out of rough endoplasmic reticulum, and they have ribosomes on them, containing ribosomes, moves to the Golgi body for final packaging. Then these transport vesicles it looks like what they're doing here, it's hard to say, but if they're going to, as it says here, they could be membrane renewal vesicles, they could be secretory vesicles if the substance is going to be spit out of the cell, to possibly undergo exocytosis. So this finishes the information for Module 2, and study, study, study. The next test will be coming up shortly. If you have any questions, always feel free to contact me.